Good morning, dear students. Today we are going to study about the electrode and its type. Okay. So first we are going to see what is electrode. What is electrode? Can anyone say what is electrode? Okay. These electrodes are employed to pick up the electrical signals of the body. Okay. You all know there will be various signals inside the body. So just to take the signals or just to measure the potential of these signals, we are using this electrode. Okay, that is, it is used to pick up the electrical signals of the body. Okay, so for recording this potential, it is necessary to provide an interface between the body and the electronic measuring apparatus. So we need to provide there will be an interface between the body and this electrodes. Okay, so this biopotential is carried out the interface function. Okay, so the electrodes what is the main use of this? The electrodes are mainly used to pick up the electrical signals of the body. Okay, this electrodes acts as a transducer. What is a transducer? Can anyone say what is a transducer? Okay, it is used to change the ionic current into electronic current. You studied in the last video, that is, this transducer is used to change this ionic current that is generated inside a body into an electronic current. Okay, the electrodes are used for measuring. What are the things we are going to measure using these electrodes? We're measuring the body signal. What are the signals? ECG. What is ECG? Electrocardiogram. Next, EEG. What is EEG? Electroencephalogram is used to measure the brain signal. Then, what is EMG? Electromyogram. What is the use of electromyogram? Just to recording the potential that is generated in the muscles, and some intercellular potentials are measured using this electrode. So this electrode is used to pick up the electrical signal of the body, and the pairs of electrodes plays a role of transducer, which converts the ionic current into electronic current, and it is also used for measuring ECG, EEG and EMG. Okay. Next, we are going to see about half cell potential. What is half cell potential? That is, this half cell potential is defined as it is the voltage that is developed at an electrode electrolyte interface. Okay, that is, there is an electrode electrolyte interface that we studied in the next slide. So if there is an voltage developed at this interface, then it is called as half cell potential. That is the potential generated between this electrode and electrolyte. Okay, so in metal solution interface, the electrode potential results from, so this electrode potential is due to the two processes, that is the passage of ions from metal into the solution. Okay, the passage of ions from metal into the solution, and also due to the combination of metallic ions in solution with the electrons in the metal to form atoms of the metal. Okay, these are the two processes. Okay, next we are going to see about electrode to electrolyte interface. We all know if there is an interface between the electrode and the electrolyte, then only we can able to measure the potential. Okay, so what is meant by electrolyte? That is, this electrolyte is nothing but it represents the body fluid that containing the ions. Okay, so the passage of the electric current from the body to an electrode can be understood by examining this electrode to electrolyte interface. Okay, that is the passage of the electric current from, that is, if there is an electric current that is generated in the body, and so that current can be understood by using this electrode to electrolyte interface. Okay, this electrolyte represents the body fluid that containing the ions. Okay, normally the net current that crosses the interface consists of three particles, that is electrons, cations and anions. Okay, that is the net current crosses the interface passing from the electrode to electrolyte. Okay, normally we know when we place an electrode there will be a current generated. Okay, so that is current crosses the interface passing from electrode to electrolyte consists of three parts, that is electrons, cations and anions. Okay, these electrons and anions will move in the direction opposite to that of the current in electrolyte, that is it moves in the opposite direction, and these cations will move in the same direction as the current. Okay, have you understood about the electrode to electrolyte interface? Okay. Next, we are going to see about perfectly polarized electrodes and perfectly non-polarized electrodes. What is perfectly polarized electrodes? Okay, it's if there is no net transfer of charge occurs across the metal electrolyte interface. That is, if there is no transfer of charge, okay, between the electrode and the electrolyte interface, then it is called perfectly polarized electrodes. And if there is an infinite exchange, okay, if there is no exchange of charge, means it is perfectly polarized, and if there is an infinite exchange of charge, means it is a perfectly non-polarized electrode. Okay, next one, artifact. What is artifact? This artifact, okay, the electrode potential is not stable and its variation constitute a source of variable noise voltage. So this we are measuring the electric potential, right? So it is stable, it is not stable, right? So if it will vary based on the input, okay, so this variation causes some noise inside this, so that noise voltage is called as artifact. Okay, it is just the noise. Okay, next one, electrode paste. Okay, the outer skin, we know the outer skin of our body. Okay, the outer skin of our body is highly non-conductive and it will not establish a good electrical contact with an electrode. Okay, it will not provide a good electrical contact with the electrode. So the skin should be washed and rubbed to remove some of the outer skin. So we all know, just to paste this electrode, we are placing this electrode on our skin, right? So when we put directly on the skin, we didn't get the contact. So we want to remove the outer skin of our body. So okay, the skin should be washed and rubbed to remove the outer skin. So this area is then coated with an electrically conductive paste called electrode paste. Then this cleaned area is coated with an electrode paste. This electrode paste decrease the impedance of the contact and also reduce the noise. So if there is any noises, means it will reduce by using this rubbing of the skin. So the electrode paste decreases the impedance of the contact and it also reduces the artifact resulting from the movement of electrode paste. Okay, generally the conductivity of the skin is directly proportional to moisture of the skin. Okay, even after the application of electrode paste, the contact impedance decreases with the increase of frequency of the signal. Okay, what are the purpose of the electrode paste? We all know the outer skin of our body is highly non-conductive, right? So the skin should be washed and rubbed to remove the outer cells, and this removed and that portion is coated with an electrically conductive paste. This paste is called electrode paste and it decrease the impedance and also reduce the artifacts. Okay, I will understand. Next we are going to see about the electrode material. Okay, the electrode paste and the body fluids can produce a battery-like actions causing ions to accumulate on the electrodes. We know this, that is just the electrode material. The electrode paste and the body fluids can produce a battery-like action causing ions to accumulate on the electrodes. Okay, so we all know the operation of the battery, right? Similarly, inside our body there will be a body fluid. So along with this body fluid, the electrode paste and the electrode can produce a battery-like action and causing the ions to accumulate on the electrodes. Such polarization of electrode can adversely affect the signal transfer. Okay, it will affect the signal transfer. To minimize the polarization, the electrode should be made of the material relatively inert to the body chemicals. Okay. Next, we are going to see about the types of electrode. What are the types of electrode? There will be three types of electrode, that are microelectrode, needle electrode, and depth electrode, body surface electrode. These are the electrodes mainly used in the human body, just for the measuring of the various signal. Okay. In this microelectrode, it is of two types, metal electrode and micropipet electrode. What are the two types of microelectrodes? Metal electrode and micropipet electrode. The next one, needle and depth electrode. Then last one, surface electrode. Okay, what is the microelectrode? This microelectrode is mainly used to measure the bioelectric potential near or within a single cell. Okay, these are called as intracellular electrodes. Okay, this microelectrodes is also called as intracellular electrodes. That is, we are placing these electrodes inside the cell. Okay, so this is called intracellular electrodes. Okay. So these are used to measure the bioelectric potential near or within a single cell. Okay. Next one, depth and needle electrode. Okay. This depth and needle electrodes are used to measure the bioelectric potential of highly localized extracellular region in brain or bioelectric potential from a specific group of muscles. Okay, this is just used to measure the sickness, that is, and the extracellular region of the brain or from specific group of muscles. Okay, that is the types of needle electrode. Okay, that you will study later. Then surface electrode. What is surface electrode? These are used to measure the voltage available from the surface of the skin and are used to sense the potential from heart, brain, okay, heart, brain and nerves. Okay, these are used to measure the potential available from the surface of the skin and are also used to sense the potential from what? Heart, brain and nerves. Okay. Next, today we are going to just see about the microelectrodes and the remaining depth and needle electrode and surface electrode we can see in the next video. Okay, and tomorrow. Okay, so microelectrodes. What is microelectrodes? Okay, this microelectrodes are divided into metallic and non-metallic. Okay, microelectrodes is divided into metallic and non-metallic. This we already seen that this microelectrodes is used to measure the electric potential within a single cell. Okay, from a single cell we are measuring this signal. So for measuring that potential from a single cell, we are using this microelectrode. This microelectrodes should have smaller diameter during the insertion of electrodes into the cell. Okay, this microelectrodes should have smaller diameter during the insertion of electrode into the cell. So that is the tip. This electrode have a small diameter tip. Okay, the tip of the electrode is very small. Okay, so when we insert this electrode inside the cell, means it will not cause any damage to the cell. Okay, during measure the potential of the cell, the micro electrode is placed within the cell and the reference electrode is placed outside the cell. Okay, we all know there will be if we want to measure the potential, we need two electrodes. Okay, one electrode we will placed inside the cell and another electrode placed outside the cell. Okay, the diameter of the tip of the micro electrodes is ranging from 0.5 to 5 microns. Okay, so what are microelectrodes? The microelectrodes are used to measure the electric potential within a single cell. Okay, it has very small diameter tip. Okay, the function of microelectrodes are potential recording. During the measuring of the potential of the cell, the microelectrode is placed within a cell and the reference electrode is placed outside the cell, and the diameter of this tip of the microelectrode is ranging from 0.5 to 5 microns. Okay, have you understand? Then in this microelectrodes, it is of two types, that is metal microelectrode and non-metallic microelectrode. This non-metallic microelectrode is also called as micropipet. Okay, what are the two types of microelectrodes? Metal microelectrodes and non-metallic microelectrodes or micropipet electrodes. First one, we are going to see about the metal microelectrode. From the figure itself, this portion is the cell. Okay, the cell consists of nucleus, cytoplasm, cell membrane. Okay, that constitute the cell. So outside the cell, we all know it's the positive energy, and inside the cell it is negative energy. So this is the microelectrode that we are placing inside the cell. Okay, this tip of this cell is have a very low diameter. It's of 0.5 to 5 microns, and there is a reference electrodes which is placed outside the cell. Okay, so this is the metal microelectrode. It is formed by electrolytically etching the tip of the fine tungsten or stainless steel wire to a fine point. This is the tungsten or a stainless steel wire to a fine point. This technique is called known as electropointing. Okay, so what is this metal microelectrode? It is formed by electrolytically etching the tip of the fine tungsten or stainless steel wire to a fine point. This technique is known as electropointing. Okay, and this metal microelectrodes are coated almost to the micro tip with an insulating material. Nearly up to the tip, it is coated with an insulating material. This shows the figure shows the insulation. Okay, insulating. To reduce the impedance, some electrolytic processing like coating the tip and developing by the photographic developer can be performed. Okay. Okay, in this figure it shows the electrode is placed on electrode, that is the metal electrode is placed inside the cell and the reference electrode is placed outside this cell. Okay, so by using these two electrodes we will measure the potential. Okay, so we can represent this metal electrolyte potential as represented as EA, and the reference electrode potential as EB, and the cell membrane potential as EC. Okay, so what about the voltage total? That is EA plus EB plus EC. Okay, that is the potential that is generated due to due to what? this metal electrode, then due to the reference electrode, and then what? Cell membrane potential. Okay. This is just the equivalent circuit of the above figure. Okay. In this, okay, just have a look. This is just shows the equivalent circuit of the above figure with RA and RB resistance of the connecting leaves, then RS resistance of the shaft of the micro electrodes. Okay, that is described here. Just have a look on this. Okay, I have to study this figure, this one, and then you have to study the equivalent circuit diagram. It is very important, these two electrodes, that is metal electrodes and micropipet electrodes. Just have a look on this, it's enough. Okay, so this is just the last equivalent circuit, approximate equivalent of the above figure. Okay, next one, non-metallic microelectrode. Okay, this non-metallic microelectrode, okay, this is also inserted inside the cell. Here also we are using two electrode. This non-metal microelectrodes or micropipet electrodes is placed, that is this micropipet electrodes, you all know that we are using pipette in chemistry lab, right? So it is similar to that. So this non-metallic microelectrodes or micropipette electrode is placed inside the cell, and this reference electrode is placed outside the cell. So this micropipette consists of a glass micropipette whose tip diameter is about 1 micrometer. What about the diameter of metal microelectrode? It is nearly 0.5 to 5 microns. Here also the diameter of this is about 1 micrometer. Okay. This micropipette, normally this micropipette is filled with an electrolyte solution. Okay, that is inside this micropipette, that is a solution known as potassium chloride, that is 3 milli of potassium chloride, which is compatible with cellular fluid. Okay, so inside this micropipette it's filled with electrolyte, usually 3 milli of potassium chloride, which is compatible with the cellular fluid. Okay, a thin flexible wire from the chloride silver, stainless steel or tungsten, okay, is inserted into the stem of the micropipette. Okay, inside the stem of the micropipette we are inserting a wire from chloride silver or stainless steel or tungsten as inserted into the stem of the micropipette. So there will be a friction generated, and this friction between the wire and the stem of the micropipette and the fluid surface tension hold the micropipette on the wire. Okay, the other end of the metal wire is mounted the rigid support, and the other end of the other end is resting on the cell. Okay, just we are using to measure this, that is this is a type of glass electrodes, and this glass electrode is inserted inside the cell. This glass electrode is filled with an electrolyte of potassium chloride, and there is a flexible wire from chloride silver or stainless steel or tungsten, which is inserted into the stem of this micro electrode. Then we will measure the variation in the potential based on this electrode and the reference electrode. Okay, this is just the equivalent circuit of the above micropipette electrode, and just have to study this. Then these are the what are the things, resistance that are used in this equivalent circuit. Just have a read on this, and this is the approximate equivalent circuit of the above figure. So this question, these two questions, okay, what are the two questions? The types of microelectrodes, it is very important Okay, so you just need to study this much. Okay, you have to study the diagrams carefully. What about the diagram? This metal electrode diagram, equivalent circuit, approximate equivalent circuit of metal electrode and also non-metallic electrode. This one, and this one. Okay, hope you understand. If you have any doubt, you can ask me. Okay, thank you.